Hey, micro friends, Dr. G here. So, just if anybody's watching this, hope to get the grades for exam 2 up today. We're going to drop exam 3 next week, and that's going to be on the different bacterial diseases. And then we'll be set for the last exam, exam 4, which is going to be on viruses and some stuff we'll be covering next week and the week after Thanksgiving. Anyway, I thought what we do today is get into some diseases caused by viruses. It's a little bit of a few things that are going to be repeat from what we've been covering in terms of viruses and things, but we're going to look at diseases of the respiratory and dermis, things like herpes virus and cold sores and stuff. So let's get into it, shall we? You know of a number of common respiratory viruses, I suspect, things like the flu. So influenza is caused by the influenza virus. It has got a phospholipid coat on it. It has protein that binds to the host receptor, and it's replicated in the genome as the viral RNA is transcribed into RNA or is maintained, and then it's packaged up and then it buds out. So this is one of those things that it's not a naked virus, and that it actually is, let's see if I can, I'm in PowerPoint here. Anyway, it actually is budding off of the cell, and so it's coated in a plasma membrane with viral proteins sticking out of it. So flu is a pretty serious issue. There have been flu pandemics in recent times. In 2009, there was a pretty significant flu epidemic. A point to note is the fatality rate in terms of comparing it with the COVID-19. The fatality rate is about 6 times less fatal. So even the really severe ones are not as nasty as COVID is. And the different kinds of flu pandemics that have come around, some that you've heard about, the 1918 flu pandemic killed a lot of people, estimates up to 100 million people. In 2009, the most recent flu pandemic, about 18,000 people died. This was a subtype known as H1N1, which has actually been around for a while. So the Spanish flu was one of the very first pandemics in recorded history in the U.S. And much like the COVID virus, it has a pretty high lethality with the elderly. Young people, very, very small infants below the age of 1 were also quite sensitive to it. And there's a little bit of a hump there in the middle, but it resembles sort of similar kinds of statistics that we see with COVID. Interesting to look at the death rate for infectious diseases, and you can see this spike here. This is the flu pandemic. We're probably going to see another one of these spikes this past year for COVID. So we know about these kinds of things. We know what damage they can do. The flu is a virus that actually mutates, something that is referred to as antigenic drift, which is a way of thinking about why the flu as an antigen, or something that is going to be lighting up the immune system. The immune system responds to antigens. Flu exhibits changes in the cell surface proteins. Consequently, the immune system isn't able to have an immune response that is going to clear the virus for every flu virus you can get. So you can get a flu vaccine, but the flu vaccine is not going to be a universal vaccine. It is designed for the specific subtype that the World Health Organization and CDC thinks is coming around, and it has to be developed back even in August or in September, in the summer, before the flu really picks up, and so they take their best guess at what they think the different strains are going to be. When they hit it, the flu vaccine is fairly effective. When they don't hit it, it's not very effective. So antigenic shift is something that occurs as the flu virus actually undergoes evolution and changes its DNA and actually changes the nucleotide sequences that produce these surface proteins. And so most of the time, the infection itself is going to be about what it normally is, but the ability of the immune system to be able to knock it back is actually what can be changing. So antivirals are an interesting study in that we've been hearing some stories about antivirals with COVID. Antivirals are not as plentiful as antibiotics, and they are not quite as effective as an antibiotic might be. Antivirals are having to focus on parts of the viral life cycle. Things like blocking the virus from getting into the cell. We have the different kinds of antivirals like amantadine that is blocking the release of the virus from this vesicle inside. And so if it can't open up into the cytoplasm, it's not going to be able to replicate. There are other kinds of antivirals, things like Tamiflu, which maybe you've heard of, which are going to slow the release of the virus from the cell. And what that means is that you have cells that are infected, but they're not releasing as much virus to infect other cells. My own personal experience, I found Tamiflu to be pretty much useless, but I think it depends on probably the severity of the case. There's a couple of things that will block the messenger RNA synthesis, but that's actually a fairly esoteric kind of thing. So it's an RNA virus, and made some mention about DNA, but it is an RNA virus, and there are changes in the nucleotide sequence that can actually change the coat protein and result in these different strains of the flu virus. Another virus that you're familiar with is the viruses that cause the common cold. Now, most common cold viruses are caused by rhinoviruses. And this is an icosahedral virus. It is a single-strand RNA. It is a plus strand. That means that RNA, when it gets into the cytoplasm, can be translated into protein. The virus is one of a bunch of members of a family, or there's a lot of members of the family. There are around 100 different rhinoviruses, and then when you combine that with other viruses, some of them even COVID viruses, that can cause the common cold, you come up with about 200 different colds that you could get in a lifetime. And pretty much once you get one of these colds, you're going to develop an immune response to it, and so you probably won't get a cold from that rhinovirus again. So if you live in a very insulated, protective spot, a very small town, and you never venture out and nobody ever comes in, there may be 30 or 40 colds that you have to get, and once you get them, you're going to be basically immune to all the cold varieties you have. Now, in the age of people traveling about, and communities and populations mixing in large cities, there are 200 or so colds, and so if you get one or two per year, well, you're going to have to live a long time before you can pick up all those different cold viruses. The symptoms are generally pretty mild, limited to the upper respiratory tract, irritation, a sore throat, mild fever, cough, and this virus is binding to receptors that are on the epithelia of the lungs and the respiratory system and the upper respiratory tract. The virus is dumping its RNA into the cytoplasm. The RNA is translated to produce viral proteins that can then package up and build the icosahedral complex. The RNA itself is replicated, and a couple of different steps are happening where you have the opposite strand being generated, and from that serves as the template for the positive strand, which is actually the genome. And that gets packaged up, and then it's released. So in this case, release is not through budding, but through the lysis of the cell. So you don't have a membrane-coated virus here. Another cold virus is the adenovirus. It's also a naked, it's a double-strand virus, and it's containing DNA, not RNA. So in this case, again, we have a receptor that binds to a portion of the virus. It is then brought in under what's known as receptor-mediated endocytosis, or the pulling into the membrane, and the vesicle that it's pulled into then allows the virus to be broken apart, and the DNA then gets out, and it is now able to get into the nucleus. And from there, it can be transcribed, turned into RNA, and you can then go through the viral life cycle. So this virus can generate more severe symptoms. It can lead to what's known as viral pneumonia. So there are a couple other things that are interesting about this virus. Is it's actually been used as a gene therapy system, specifically for use in cystic fibrosis, where cystic fibrosis is a mutation in a chloride transporter. It's most affected in the lungs. There are other parts of the body that need that chloride transporter, but you can kind of get around that. But in the lungs, though, because the chloride transporter is not working properly, you end up with real thick, heavy mucus. And what scientists, investigators have done is put in a functioning CF transporter into the viral genome. You infect somebody, they huff the genetically modified virus. It infects the lining of the lungs, as all good cold viruses should do, and then it provides a new copy of that chloride transporter, the CF transporter gene. And for a period of time, you have lung cells that are producing the appropriate chloride transporter, and it works. The difficulty is that because you're not getting in, the virus doesn't get into the stem cells, they're going to constantly be regenerating the lungs. You're simply infecting those cells that are on the outer surface of the lungs, and those cells over time, 6 weeks or so, slough off, and you have new cells coming. And so this treatment has to be renewed every 6 months or so, or every 6 weeks or so. Another one of the potential issues is that it is designed as a gene vector. It's designed to be non-infectious, so it can't replicate. But if you've ever watched Jurassic Park, the idea being that you can have these systems in place that'll inhibit replication, but if they get mutated, things happen. You got a lot of viruses there, so the chance of mutation is very much going to happen. You can begin having people that are exhaling virus that is genetically modified, shouldn't hurt anybody, but the possibility of something going wrong is actually quite, is a legitimate point. So, it's a really cool idea, but it really hasn't come into widespread use. Another virus perhaps you have not heard of is called respiratory syncytial virus. And it's a virus that's related to the mumps virus. Most kids are going to be infected with it by 3 years of age. Because of the virus and the way it interacts with the immune system, it's not something that generates a real strong immune response, and so you can have it again. You get mild flu symptoms. You do have the possibility of significant lung inflammation, and that is the main concern. So here's an electron micrograph showing the virus budding from the cell. So this is one of those that is not naked. It actually has a phospholipid bilayer around it. So here's a coronavirus that for a time was the big coronavirus people talked about, which is the severe acute respiratory syndrome virus, known as the SARS virus. It was discovered in an outbreak in China. It killed 774 people worldwide, really high mortality rate, but it kind of went away. The origin is one of these zoonotic type diseases in which there is a wild organism, a bat, that passes it to a civet, that then passed it to a human. What's a civet? I don't know, sort of a mink-looking guy, I think. And this gets into the lungs. You end up having basic cough, flu or cold symptoms. It can then progress, though, into breathing difficulties, pneumonia, and from there, that can trigger lung failure, which of course will lead to death. Oops. So SARS spread from China, and in the same way that COVID-19 has spread. And in this case, with SARS, it kind of it wasn't as infectious, and so it didn't go as far as the COVID has been going. COVID, as it turns out, is actually quite infectious, more infectious than the flu even. And so while the symptoms are not quite as severe for most people, it is spreading around a lot. And as I say right now, here in early November, the COVID infection rate is absolutely skyrocketing, and we're having a difficult time trying to actually control this. And at this point, waiting on a vaccine is probably not the sort of tactic we should take. Really what we need to do is simply practice the masking and social distancing. And if this were being practiced in a really serious way, we could bring the virus load down actually fairly quickly. So there are a number of viruses that are related to skin diseases. A real common one is the herpes virus. It's known as a cytomegalovirus. It is a membrane-coated virus. It's got its genome packed inside the nucleocapsid. And there are a number of different herpes virus types. They cause everything from herpes simplex 1, which is a cold sore that many people are infected with, to chickenpox and shingles. That's the herpes zoster virus. HHV3, as is known, is the human herpes virus. Another one that's not pretty common is Kaposi sarcoma. This is caused by the HHV8 virus. This is something that is seen primarily in the severely immune compromised, and it was first observed with people that were infected with a virus that causes AIDS, which we'll be getting into next week. And this virus begins to attack the immune system, and the T cells are a part of the immune system. Once the immune system is not working very well, people begin to exhibit this kind of disease. One of the things this means is that for a reasonably healthy immune system, the Kaposi sarcoma virus is actually very easily removed by the immune system, and not exactly known why or how, but our immune systems are very effective at removing Kaposi sarcoma, or the virus that causes it. So there are a number of different human herpes viruses, and we certainly don't need to memorize all of them, but just looking at some of the things they cause. So HHV1 is oral herpes. HHV2 is a different virus, causes genital herpes. HHV3 is what causes chickenpox, and then, as we're about to see, this virus actually is able to isolate itself in neurons, and it can come out later as shingles. And so this is the same thing that's happening with oral herpes, is that you can't really get rid of it. It's actually going to be silent and a reservoir in the neurons. Other herpes viruses, HHV5 is one of the main causes of mononucleosis. The other main cause of mononucleosis is the Epstein-Barr virus. Epstein-Barr virus actually has a number of different sorts of associations with things like chronic fatigue syndrome and things. So let's see. HHV6A is causing something called roseola, or rash, with a mild fever. 6B and 7 really have no known pathology. They're not doing much to you. We know they're around, but they don't really seem to do much. Okay, so a wide variety of viruses and viral types. One of the things, though, with viruses in general is that there's not great treatments. We have antivirals, but primarily what you do with viruses is you treat the symptoms. So with herpes, we have fusion. The nucleic acid gets into the nucleus. The nucleus is then going to be, or the DNA is then going to be transcribed into RNA using RNA polymerase. And from there, viral DNA can make its way back out. The RNA is going to be used to generate all the proteins necessary for packaging up the virus, and the virus then leaves by a budding mechanism again, which is why it has that membrane-bound coating. Now, one of the things that's interesting about herpes simplex, we actually alluded to this in our previous discussion, is that it can actually end up getting into the cells of the neurons and exist as what's known as an episome. It is essentially a non-chromosomal piece of genetic material that is maintained. And so you have these latent infections in the neurons, and the reactivation is going to generate a large amount of viral replication, and that is going to then lead to these open dermal sores, and it then goes back into latency. So all the herpes viruses have the ability to establish these latent infections, but the target cells can end up being different. And so one of the things that happens is that because they're tracking on neurons, you end up with a side of the face that might have shingles, or a side of the face that is going to have a cold sore. And they kind of tend to, because they're in these certain neurons, they tend to actually end up in the same place over and over again. A couple more viruses that we have heard of. Just going to go through the viruses that are part of the measles, mumps, rubella vaccine. The measles is a enveloped RNA virus. It generates a rash and a very high fever, fairly high mortality in unvaccinated children. Nowadays in the USA, it's actually quite rare. The vaccine works quite well, so we end up having both the herd immunity and the actual virus is fairly rare in the population anyway. Measles generally remain fairly low, but because of decrease sometimes in compliance with vaccination, there are the occasional measles outbreaks. So last year there was 1,271 cases, actually fairly low, but preventable. So here's the basic measles life cycle. It is an RNA virus. So the RNA is being brought into the cytoplasm. The RNA itself is going to be synthesized and to produce more RNA. The capsid proteins then can be translated. The RNA then is packaged back up. And this is a little interesting point. The virus RNA is what's known as a negative strand, meaning that it is not actually the template for translation. It has to be converted. That is, the RNA is coded again on the other strand to make what's called a positive strand, and that can actually generate proteins and serve as a template for proteins. Just one of the things that viruses do, all kinds of weird things. Mumps is a paramyxovirus. It is enveloped. It is also a negative single-strand RNA virus. So that means that the viral complex has to come in with some proteins that can take that RNA and turn it into a positive strand. That is, they actually have to transcribe it again and polymerize RNA from the other strand. It's a fairly mild disease. Most, at least 20% asymptomatic. The primary symptom is going to be swelling of the salivary glands, and that gives the characteristic swollen mumps face. You also have fever, headache, you're tired. Testicular inflammation, that sounds unpleasant. It actually can lead to sterility because of that. The complications are fairly rare, but they can be somewhat severe. So the cases of mumps is fairly low, but it does come up. Vaccines work. So let's see. Rubella is the third part of the MMR vaccine, and this is also known as German measles. Now, this is a plus strand enveloped virus. Now, what this means is a plus strand, when the RNA gets in, is actually able to be translated directly. Main concern is in pregnant women. They can pass it on to the fetus, and that can cause a range of pretty severe abnormalities. So, rubella really just isn't present in the population. Occasionally, infections are seen primarily from people coming from outside the U.S. So, just a little discussion about vaccines in general here. The MMR vaccine is quite effective. So we see here that prior to the vaccine being licensed, we had thousands, hundreds of thousands of cases of measles. When the vaccine comes in, the cases plummet. Now, over time, a second dose was recommended, which is sort of a booster shot, and that really keeps the numbers down very, very low. Let's see. So, measles and rubella, they were pretty much eliminated. There are no continuous infections. Mumps is very low. The vaccine for mumps is not quite as good. So the MMR is a combination of these three different viral antigens. Another thing that you perhaps have heard of is the association with autism, learning disabilities, with the MMR vaccine. And this top graph is been a way of trying to say that the MMR is responsible for increase in autism. But it turns out that there really is no good correlation with the MMR vaccine. That part of what happens is that autism is been more easily identified now, and it's going to come about around age 3 or so, not long after kids had their second dose of the vaccine. So it's associative. It is not causative, and there is no evidence to say that it is. So, there. Hey, as far as skin goes, we've mentioned a little bit about the human papillomavirus. This is one of the viruses that can cause things like genital warts. So, there are a couple of different kinds of warts around. Different strains of the virus cause warts in different places. Most are benign, but some of them can cause a variety of cancers. So most of the time, a wart, if you let it go long enough, just let it sit, it will resolve on its own. There are always going to be new infections each year. The best treatment for HPV is, of course, the HPV vaccine. So, HPV is associated with a fair number of ovarian and or cervical cancers. So, cervical screenings are primarily done by Pap smear, which is actually a very old-fashioned way of looking at cells and looking at the membrane in those cells. And abnormal cells then get diagnosed as potentially being cancerous, and then you have a biopsy and a little bit more study. Very often, these cervical cancers are associated with cells that have been infected with the virus. So, estimated that at least of those kinds of cervical cancers, 90% of them could probably be eliminated with a vaccine to HPV. Let's see. A couple of the viruses that you perhaps know of is some of the different pox viruses, things like chicken pox, smallpox, cowpox. This has been eradicated. Basically, there is no more pox virus on the planet, other than in places that actually hold viral DNA or viral particles as part of a disease storage facility and things. This virus is a DNA virus. It's got a fairly large genome, actually, for viruses, 200,000 nucleotides. It is cytoplasmic, and it actually is able to do all of its own replication in the cytoplasm. It is one that the vaccine works very, very well and essentially eliminated it. And so now I had a smallpox vaccine when I was 5, 6. But if you were born probably past 1980 or so, you don't have a smallpox vaccine. So none of you have had the smallpox vaccine unless you from some countries continued the smallpox vaccine longer than the U.S. did in Asia and some parts of Africa. But smallpox has been basically eradicated. Hey, so that's what we have to say about that. We'll get into some more discussions about viruses in the blood and a few other different kinds of viruses next time we meet. And that's all I know. Thanks for your attention, and I'll talk to you soon. Bye.